Hello students, in this video I'm going to explain about the biosynthesis of purine and pyrimidines. Okay, what is biosynthesis? It is nothing but it is a multiple step or enzyme catalyzed process where substrates are converted into more complex products in living organism. Okay, in biosynthesis, simple compounds are modified, converted into other compounds, or joined together to form macromolecule. Okay, so this process often consists of metabolic pathways. Okay, so many processes like nucleic acid metabolism or carbohydrate metabolism or like this, so many metabolisms are carried out in our body. Okay, so nucleic acids are also formed in this way. Okay, so you know that nucleic acids are the polymer of nucleotide. It is like ATP, GTP, UTP. So these are required to provide energy. Okay, adenosine triphosphate, guanosine triphosphate, and uracil triphosphate are required to provide the energy, and CTP, cytosine triphosphate, it is a source of energy in lipid metabolism, and AMP, it is needed as a part of the coenzyme like NAD and coenzyme E. Okay, our body it is able to synthesize nucleotides and also metabolize them to be reused. Nucleotide synthesis it is an anabolic process involving the joining of phosphate, pentose sugar, and a nitrogenous base. Okay, the phosphate group it is a common to both DNA and RNA, but they differ in the sugar and with respect to only one nitrogen base. These basic things you're already known. Okay, RNA it has a ribose sugar and DNA has a deoxyribose sugar, and mainly nucleotides are divided into purine and pyrimidines. Purines are adenine and guanine. Pyrimidines are thymine, uracil, and cytosine. Okay, and the purine are built upon a pre-existing ribose 5 phosphate, and liver it is a major site of purine nucleotide synthesis. Erythrocyte, polymorphonucleoleucocytes, and brain cannot produce the purines. Okay, so main precursor for the nucleic acid synthesis it is a phosphoribosyl pyrophosphate, or it is abbreviated as PRPP, which donates the ribose and phosphate necessary to make a nucleotide. Okay, so let us discuss the different steps or the different methods involved in the nucleic acid synthesis. Okay, so there are mainly two pathways for the synthesis of nucleotides. The first one it is the de novo synthesis. Here biochemical pathway where nucleotides are synthesized from new simple precursor molecules. Okay, so here the de novo synthesis, that major mechanism requires some of the precursors to produces the nucleic acid. Okay, and another major pathway it is a salvage pathway. It is used to recover bases and nucleotides formed during degradation of RNA and DNA. Okay, so let us discuss the pathways. Okay, see here nucleotide biosynthesis mainly include two pathways, de novo synthesis and salvage pathways, and both purine and pyrimidine it is also synthesized by the de novo synthesis, and purine and pyrimidine also synthesized by the salvage pathway. Okay, so in de novo synthesis purine synthesis mainly required the formation of IMP. Okay, and pyrimidine it is by UMP and DUMP. That all steps I will explain in the next slide. Okay, and in salvage pathway also both purine and pyrimidines are produced. Okay, then let us first discuss about the de novo synthesis of purine. Okay, and here this image shows, see you can see one image in this slide. Okay, so it is a source of different atoms in a purine skeleton. Okay, N1 it is from amino group of aspartate, C2 and C8 from the formate, N3 and N9 from amide group of glutamine, C4, C5 and N7 from glycine, C6 from bicarbonate. Thus, aspartate, formate, glutamine, glycine, and bicarbonate act as a building block of the purine synthesis. Okay. So purine, you know that it is adenine and guanine. Okay, it is derived from inosine 5' monophosphate. Okay, so it is mainly start with that GMP and AMP, or adenosine monophosphate. It is mainly started with the formation of inosine 5 prime monophosphate. So here inosine 5 prime monophosphate, it is a main precursor for synthesis of AMP and GMP. Okay, let us discuss the steps involved. Ribose 5-phosphate of a carbohydrate metabolism is the starting material for purine nucleotide synthesis. Okay, it reacts with the ATP to form phosphoribosyl pyrophosphate, or it is PRPP, and glutamine transfer its amide nitrogen to PRPP to replace the pyrophosphate and produces the pyro 5 phosphoribosyl amine. Okay, and it is catalyzed by the enzyme PRPP glutamyl amidotransferase. Okay, this reaction is committed. Okay, then the next step, it is phosphoribosyl amine react with glycine in the presence of ATP to form glycine amide ribosyl 5 phosphate, or glycine amide ribotide GAR. It is catalyzed by the enzyme synthetase. Okay, then N10 formyl tetrahydrofolate donates the formyl group, or the product formed is formyl glycine amide ribosyl 5 phosphate. It is catalyzed by formyl transferase, and glutamine transfer the second amino group to produce formyl glycine amide ribosyl 5 phosphate, and it is also catalyzed by the enzyme synthetase. Okay, then the imidazole ring of the purine is closed in an ATP dependent reaction to yield 5 prime amino imidazole ribosyl 5 phosphate. It is catalyzed by the synthetase. Incorporation of carbon dioxide occurs to yield amino imidazole carboxylate ribosyl 5 phosphate, and it is catalyzed by the enzyme carboxylase, and it does not require vitamin biotin or ATP. Okay, then further step include aspartate. So it will undergo the condensation with amino imidazole carboxylate ribosyl 5 phosphate to form amino idazole 4-succinyl carboxamide ribosyl 5 phosphate, and it is catalyzed by the enzyme synthetase. Okay, so here it is the steps involved in Denovo synthesis of purine. Okay, so here the substrate required, it is the ribose 5 phosphate, glycine, glutamine, water, ATP, carbon dioxide, and aspartate. Okay, again I am repeating, in Denovo pathway of purine, that is the synthesis of AMP and GMP. Okay, so that requires a substrate, or the major precursor for that particular reaction. It is ribose 5 phosphate, glycine, glutamine, water, ATP, carbon dioxide, and aspartate. Okay, and the product formed, it is a GMP, AMP, glutamate, fumarate, and water. Okay, so here the first step, okay, it is synthesized on the ribose moiety. Here 5 prime phosphoribosyl 1 prime pyrophosphate, which provide the ribose moiety that react with the glutamine to form phosphoribosyl amine. This first step in purine biosynthesis produce N9 of the purine ring, and it is inhibited by the AMP and GMP. Okay, then the entire glycine molecule, it is added to the growing purine precursor. Then C8, it is added by formyl FH4 N3 by glutamine, and C6 by carbon dioxide, and N1 by aspartate, and C2 by the formyl FH4. Okay, IMP, which contains the base hypoxanthine, and it is generated. Then IMP is cleaved in the liver, and its free base or nucleoside travels to various tissues when it is reconverted into the nucleotide. Okay, then IMP, it is the precursor for both AMP and GMP. Each product by feedback inhibition regulate its own synthesis from the IMP branch point, as well as inhibits the initial steps in the pathways. Okay, then AMP GMP can be phosphorylated to the triphosphate level. The nucleotide triphosphate ATP and GTP can be used for energy requiring process or for the RNA synthesis. Okay, then the reduction of ribose moiety or deoxyribose occurs at the diphosphate level and is catalyzed by ribonucleotide reductase, which requires the protein and thioredoxin. After the diphosphates are phosphorylated, DATP and DJTP can be used for DNA synthesis. Purine bases can be salvaged and converted between free bases, nucleotides and nucleosides by the series of reaction. Okay, so here the first step, it is a precursor, it is a ribosyl 5 phosphate that undergo the series of reaction by the addition of glutamine. Okay, and ultimately there is a formation of inosine monophosphate, that is IMP. Okay, so then the synthesis of AMP and GMP takes place from the IMP. Okay, so first it is synthesis of AMP from inosine monophosphate. Okay, so it is the immediate precursor for the formation of AMP and GMP. So here I already told that aspartate, it is condenses with the IMP in the presence of GTP to produces adenyl succinate, which on cleavage forms the AMP. Okay, then formation of guanosine monophosphate. Okay, how guanosine is formed from IMP. Here IMP undergoes NAD plus dependent dehydrogenation to form xanthosine monophosphate. Glutamine then transfers amide nitrogen to xanthosine monophosphate to produces the GMP. Then 6-mercaptopurine, it is an inhibitor of the synthesis of AMP and GMP. It acts on the enzyme adenyl succinase of AMP pathway and IMP dehydrogenase of GMP pathway. Okay, see here the synthesis of AMP and GMP from the inosine monophosphate. Okay, aspartate plus GTP adenyl succinate synthetase, and here GTP will condense to form GDP. So here is the formation of adenosyl succinate. Okay, so IMP combined with the aspartate. Okay, then adenosyl succinate, so undergo the condensation. So adenyl succinate, it is eliminated from that. Okay, and there will be a formation of AMP, that is nothing but adenosine monophosphate. Okay, and another, it is the formation of GMP. Okay, here also IMP, it is a precursor. Okay, so here it is an enzyme NAD plus it is required, and the enzyme NAD plus, it is a coenzyme required for that reaction. Okay, and enzyme required, it is IMP dehydrogenase. Okay, NAD plus, it is condensed into the NADH plus H plus. So here the intermediate product formed, it is the xanthosine monophosphate. Okay, and the another enzyme required, it is a GMP synthetase. Okay, so here glutamate and AMP plus inorganic phosphate, it is formed by the by formation of the GMP. Okay, so this is about the formation of purine, that is adenine and guanine from Denovo synthesis. Okay, so here the main precursor substrate or precursor, it is a ribose 5 phosphate and glycine and glutamine. So these are undergo the series of reaction, and it is reaction is in the intermediate product, it is the IMP, and from the IMP the two purines, that is AMP and GMP is formed. Okay, so that is the one pathway of the formation of the purine. Okay, and the next pathway, it is the salvage pathway of the purine and purine. Okay, so here in a salvage pathway, so here it is a pathway in which nucleotides are synthesized from the intermediate in the degradative pathway for nucleotide. Purine synthesis via salvage pathway occurs in all the tissues. It is especially important in the brain and bone marrow. Okay, here the substrate required, it is a hypoxanthine and enzyme PRPP, guanine and adenine, and product formed it is the GMP, AMP and IMP. Okay, so here adenine, so adenine it is a precursor, or it is already formed and it is degraded as the some of the metabolic product. Okay, so from that it is the formation of the AMP. Okay, so there is a enzyme adenine phosphoribosyl transferase. Okay, and the PRPP also involved in this. Okay, and adenine it is converted into the AMP. Okay, and another guanine, so here also enzyme required, it is the hypoxanthine guanine phosphoribosyl transferase. Okay, and here also PRPP converted back into the PPI, and the GMP it is formed. Okay, then hypoxanthine also converted into the IMP, and from IMP AMP and GMP is formed. Okay, for the conversion of hypoxanthine into IMP, so that is required enzyme, it is hypoxanthine guanine phosphoribosyl transferase. Okay, so here it is, this is about the salvage pathway of the um, synthesis of purine. Okay, so here some of the inhibitors of purine biosynthesis, that is folic acid, it is essential for the synthesis of purine nucleotides. Then sulfonamides are the structural analogues of para-amino-benzoic acid, and these sulfur drugs can inhibit the synthesis of folic acid by microorganism. This indirectly reduces the synthesis of purine and nucleic acid. Okay, the structural analogues of folic acid, it is methotrexate, used to control the cancer. Okay, then another it is azathioprine, it is the glutamine antagonist and inhibit reactions involving glutamine. And other synthetic nucleotide analogues used as anti-cancer agents are 6-theoguanine and 8-azaguanine. Okay, so this is about the two pathways, Denovo pathway and salvage pathway, regarding to the synthesis of purine. Okay, and next coming slide I am going to explain about the synthesis or biosynthesis or mechanism of synthesis of pyramidine. So this also involves the two pathway, that is de novo and the another it is the salvage pathway. So first let us discuss about the de novo synthesis of pyramidine. Okay, so here biosynthesis of pyramidine, it is simple than that of the purine. Okay, following diagram shows the sources of different atoms in a pyramidine skeleton. So you can see the diagram, it is a pyramidine nucleus. Okay, so that also consists bicarbonate, aspartate and glutamine amide. Okay, so N1, C6, C5 and C4 from aspartate, N3 from glutamine, C2 from bicarbonate. Okay, in pyrimidine nucleotide synthesis, the ring is completed before being linked to ribose 5-phosphate. Okay, and so let us discuss the steps involved in the formation of uridine monophosphate. Okay, how the IMP is important in formation or synthesis of purine. Okay, and here the in pyrimidine synthesis, the formation of UMP, it is important, that is uridine monophosphate. Okay, so uridine monophosphate is also act as the precursor of the CMP. CMP it is cytosine monophosphate, and UMP it is synthesized in 6 step. Okay, so formation of UMP, so it is followed by, or it is completed by having 6 major steps. Okay, so first step, it is a synthesis of carbonyl phosphate with the hydrolysis of 2 ATP, bicarbonate and amide nitrogen of glutamine combine to form carbonyl phosphate. Okay, so in the first step, glutamate, and it requires the ADP plus inorganic phosphate. Okay, so first it required 2 ATP molecule and bicarbonate and glutamine and water. Okay, and condensation of glutamine to glutamate, and ATP is dephosphorylated into the ADP and inorganic phosphate, and here the enzyme involved is carbonyl phosphate synthesis second. Okay, so there will be the formation of carbonyl phosphate. Okay, and in the step 2, that is a synthesis of carbonyl aspartate. Okay, so carbonyl phosphate react with the aspartate to yield carbonyl aspartate. Okay, so here carbonyl phosphate is formed in step 1. Okay, so that react with the aspartate. So here the enzyme aspartate transcarbamylase ATP CS. Okay, so that will involve the synthesis of carbonyl aspartate. Okay, next third step, it is the ring closure and dihydrorotate or dihydrorotate formation. Okay, here by the elimination, or here the reaction, it is the condensation reaction. The carbonyl aspartate, it is converted to a ring compound that is called as a dihydrorotate. Okay, one molecule of water is eliminated in step 3. Okay, then the next step, it is the oxidation of dihydrorotate. So dihydrorotate is dehydrogenated to form orotate. Okay, orotate. Then step 5, it is the acquisition of the ribose phosphate moiety. Orotate react with the PRPP to produce orotidine 5' monophosphate, or it is called as a OMP. And in step 6, decarboxylation to form UMP. So OMP undergoes decarboxylation to form the UMP. Okay, so by this way UMP is formed. Okay, so mainly it involves the 6 steps. Okay, next the synthesis of UTP. Okay, so it is uracil triphosphate. UMP, it is converted to UTP in two-step kinase reaction. Two ATP molecules are required. Okay, so here UMP by the help of ATP, so it will converted into the UDP, and ATP, it is dephosphorylated to ADP. So UDP, it is converted into the UTP, and it is a step where the UTP is formed. Okay, then synthesis of CTP, another pyramidine. The first pyramidine, it is uracil, and next pyramidine, it is a cytosine. Okay, cytosine triphosphate, it is synthesized by the amination of the UTP. So first UMP is formed. Okay, from the UMP UTP is formed. Okay,